Good afternoon, everybody. On behalf of the Lupus Foundation of Minnesota, I want to thank everyone for joining us today. My name is Cheryl Como, and I'm Vice President of Client Services and Social Worker for the Lupus Foundation. I'll be your host for today's webinar, Interferon in Systemic Lupus, Friend or Foe. Before I introduce our speaker, I want to talk briefly about today's program. Today's webinar will run up to 1:15 p.m. You'll see a dialog box on your screen. Please type your questions and comments in this area throughout the webinar period. Time will be provided after Dr. Erikson's presentation for Q&A as well. Today's webinar goals include to discuss how the University of Minnesota researchers have linked interferon to lupus and how lupus risk genes might control the levels and function of interferon. Review the progress to produce new treatments that target interferon, and describe a new study that looks at how PTPN22, one lupus risk gene, affects interferon-producing capacity of lupus patients. Just as a slight warning, there may be technical difficulties that occur on my end, but I will make every effort to ensure that this learning opportunity goes uninterrupted. So whether you're new to webinars or a seasoned veteran, here are some tips. On the screen, you see the GoToWebinar support services number. In the event that you experience technical difficulties on your end, please jot this number down along with the webinar ID. There'll be a number of prompts to choose. Please choose in session. You will get faster service that way. Audio is through headphones or the speakers that are attached or built into your computer. To facilitate sound quality, be in a quiet area and adjust your sound as necessary. During the webinar, everyone is on mute status except for the presenter. Please type your questions in the box on your screen. We will take questions throughout the webinar, so please feel free to just keep adding them. Presentation slides will be viewed from your computer screen. If you're having technical difficulties, again, please contact the support number. Keep in mind that the quality of your connection can interfere with slide transition and sound quality, and at times, weather can impact connection as well. At the conclusion of the webinar, a survey will arrive via email. We hope that you take a few minutes to complete it. And finally, if you require a certificate of attendance, my email will be posted on the last slide, so please contact me and one will be sent to you. So without further delay, I would like to introduce our speaker, Dr. Erik Peterson. Dr. Peterson joined the University of Minnesota Division of Rheumatic and Autoimmune Diseases in July 2002. He is a member of the Interdisciplinary Center for Immunology and its Autoimmunity Program, and a member of the Cancer Center. He is an immunologist with a scientific interest in those molecules that regulate the development and function of the immune system and promote the development of autoimmunity. He also practices general rheumatology and participates in resident and fellows teaching. Dr. Peterson has strong interests in the molecular underpinnings of autoimmune diseases, including rheumatoid arthritis, lupus, and myositis. His laboratory utilizes genetic, biochemical, and primary human sample-based approaches to investigating the mechanisms whereby recently identified risk genes predisposed to development of autoimmune disease. Dr. Peterson's group recently identified a role for PTPN22, which is a potent risk gene for many autoimmune diseases, in the promotion of toll-like receptor signaling and type 1 interferon production. He is currently investigating the role of PTPN22 and myeloid cell functions in systemic lupus and in response to immunization, as well as characterizing the molecular mechanism of PTPN22 promotion of type 1 interferon signals. So without further delay, welcome Dr. Peterson. The floor is yours.
Cheryl, thank you very much for the introduction, and thanks to you and the staff of the Lupus Foundation of Minnesota for arranging this forum for me to conduct what I hope will be an informal presentation with question and answers all along the way. I want to start out by saying that I'm directing this presentation towards a person who may have an interest but no specialized medical training in lupus or lupus research. And so if you hear me using jargon or terms that you don't understand, please electronically raise your hand, and I'll see to it that I explain everything as best I can. I'm, as Cheryl said, a researcher at the University of Minnesota, where I also practice rheumatology. And for a number of years, I've been very excited by and interested in pursuing research that has, I think, begun to give us some clues about the cause of an illness that I see frequently in the clinic, systemic lupus erythematosus. So today, I'd like to spend some time, first of all, giving you a little bit of a refresher background about lupus itself, and then take you into a brief historical tour concerning one of the molecules or proteins in the blood that have recently been identified as being strongly associated with lupus. Now, one of the thoughts about this molecule, type 1 interferon, is that it might actually cause disease to be worse in lupus. But there's growing evidence that the type 1 interferon that I'll describe might also, paradoxically, ameliorate inflammation and disease severity in lupus. And finally, I'm going to talk to you a little bit about an exciting new study of interferon in lupus. And this is a study that was recently funded by the Lupus Foundation of Minnesota. I am the principal investigator on the study. I'm very fortunate to be aligned with the Lupus Foundation to be able to conduct this research.
Okay, Dr. Peterson has a question for everybody. I'm going to launch a poll. So if you could respond to the poll, we're just going to start out with a quick little test.
Please don't feel oppressed by
No.
terminology here. This is just to stimulate your thinking about some of the content of our discussion.
Well, everybody here has a great grasp. They chose lymphocyte activities against self. That particular, but 83% have chosen that.
Excellent.
That answer.
Did anybody choose infection fighting?
Yep, about 13%.
Boosted by the capacity to drive to work?
Yes, about 13%.
Okay, that was my joke.
That was your joke.
I can hear everybody laughing loudly. Yes. So I'm trying to advance this slide now.
Okay.
And I hit the down arrow, and
Okay, here, wait a second.
there we go.
I lost everything. I'm sorry.
Okay. Can you still hear me, Cheryl?
Yes, I can.
All right. So I'm now looking at a slide entitled Systemic Lupus Erythematosus. I hope everyone else is on the same slide.
Yes.
And this just has a few broad overview points about lupus so that we're on the same page. Lupus is defined as a chronic autoimmune condition that has a very strong heritable or genetic component. However, genes aren't everything. It's almost certainly true that environmental factors such as infections with viruses or toxins, chemicals, may trigger the disease. It is known to be a very prolific disorder in that it affects many parts of the body and organ systems, sometimes simultaneously and sometimes serially. It's characterized by waxing and waning, flaring and remitting disease activity often through months and years. And this characteristic of the disorder prompts clinicians to usually perform frequent monitoring of patients and tinker with or adjust therapy frequently to counter these flares of disease activity. One of the prime features or hallmark features of the autoimmunity in systemic lupus is the presence of unique blood proteins referred to as autoantibodies, that is, antibodies that are reactive with self. And these are thought to be important for the actual process of organ and tissue damage in chronic lupus. Some of the names you'll hear bandied about when persons are discussing autoantibodies in lupus are anti-nuclear antibodies, ANA for short, anti-double-stranded DNA or anti-DNA antibodies, anti-Rho, anti-La. These are all names of antibodies that have been associated strongly with the tissue injury that occurs in systemic lupus. Next slide.
Oh. These are some of the technical difficulties I was talking about. I seem to have lost my connection. Can you keep talking, Doctor?
Sure. So I wanted to just make a few comments about the burden of systemic lupus in our society. I'm just bringing up my own copy of the presentation on the computer so that I can continue. What are the attendees viewing right now, Cheryl?
Right now, that's what I have lost. So,
okay, so there's no visual right now for these facts about lupus. About 1.5 million Americans are thought to carry the diagnosis. This is a burden that's heavily tilted against women, since about 90% of lupus patients are women. The majority of women affected by the disease suffer with it during their working and childbearing productive years. And it is difficult to deal with. 2 out of 3 lupus sufferers report partial or complete loss of their income due to the condition, and up to 1/4 of lupus patients are fully disabled. The cost of caring for people with lupus to the healthcare system in the United States is approximately $8,000 to $10,000 per year per patient. So there's a big societal and individual burden associated with systemic lupus.
we are back.
All
After
right,
you
let me
go right through the
see
slide.
where connected Google. I am not seeing the slides on my screen anymore. Just a second. Oh, gone back.
There you go.
Now I'm looking at a slide, and hope everyone else is as well, entitled Lupus SLE Diagnosis.
Yes.
It's a difficult disease to diagnose. In the past years, a frequently cited feature or fact about lupus is that practitioners, even experienced practitioners, frequently don't, I shouldn't say miss, but do not tumble to a diagnosis of lupus when confronted with what are often nonspecific symptoms for sometimes years in individual patients. We make the diagnosis by putting together a collection of symptoms, signs, and selected laboratory tests when they form a pattern that is consistent with systemic lupus. Some of the criteria that have been established by the American College of Rheumatology for making the diagnosis include a malar rash, that is a rash that occurs over the cheeks of the face or the forehead, sometimes the upper chest and forearms, recurrent oral ulcerations, sometimes painful in the soft palate or soft part of the upper mouth and the nasal passageways, a phenomenon of photosensitivity, meaning there's sensitivity to light frequently patients with lupus, after even mild exposure to direct sunlight, report symptoms of nausea, headache, or just feeling sick like they have the flu. That is photosensitivity. Discoid rash is another specialized, uncommon form of rash that is found in patients with lupus. Other criteria outside the skin include inflammation of the body cavities, like the lining of the lungs and the lining of the cavity of the gastrointestinal tract. Additionally, arthritis, particularly inflammatory arthritis affecting small joints of hands, feet, and bigger joints as well, knees, elbows, shoulders, are characteristic of lupus. A big organ involved frequently in lupus is the kidney. About 50 to 60% of lupus patients at some point during the course of their condition will develop kidney disease. And this can range from mild loss of protein into the urine to complete organ failure and need for transplantation or dialysis. Fortunately, infrequently, the central nervous system, including the brain and the spinal cord, can be involved with lupus as well. In the laboratory, we find some characteristic but non-specific findings, including low white and red blood cell counts and platelet counts, and the autoantibodies that I described on an earlier slide. Next slide, please. There aren't many treatments for lupus, and many of the treatments that we apply in the clinic are not, quote, FDA approved, unquote, because we don't have hard and fast evidence that they alter the natural history of the disease. But I wanted to just briefly review that the fact that there are treatments and that, in fact, the prognosis and the course of lupus for the vast majority of people with the condition has improved in the past quarter century due to these treatments. One of the cornerstones of treatment is drugs that are in the chemical class of cortisone. And many listeners may be familiar with the drug prednisone, which is a synthetic, souped-up form of cortisone. Secondly, a drug called hydroxychloroquine, a brand name Plaquenil, is an immune modulating medication, and this is a drug that has been used frequently and effectively for many years to help especially patients with arthritis and skin manifestations or skin features of lupus. A new kid on the block in the treatment arena is the drug Benlysta, that's the brand name. The chemical name is belimumab. This is a so-called biologic treatment, meaning that it's a drug that cannot be given by mouth like the other two on this slide. It cannot be given by mouth because this medication is akin to insulin. When given to diabetics, cannot be taken by mouth and must be injected under the skin. The reason for that is this drug is actually a protein, and proteins would be degraded in the acidic environment of the stomach. So we have to avoid the gastrointestinal tract when giving this drug, Benlysta. Benlysta has been approved now for about 2 years and has been utilized in lupus patients whose arthritis and skin disease in particular has been difficult to manage with cortisone or Plaquenil or some of the other immunosuppressive drugs that we commonly apply. So when we put together the information about lupus in this current day and age, from one point of view, it's a bit daunting. We still have no known cause of the disease. We do not have a cure. This is a chronic condition. The current treatments are not adequate to protect fully the organs and tissues from the effects of long-standing inflammation and autoimmune disease. And as we've discussed, the personal and the financial burden of the disease in our society is quite heavy. So I think we might be daunted to the point that the dinosaurs were about 63 million years ago in this cartoon from Gary Larson, where the head of the dinosaur convention is getting up and saying, the picture's pretty bleak, gentlemen. The world's climate is changing, the mammals are taking over, and we all have a brain about the size of a walnut. I don't think it's quite as grim as that, and I'm excited to say that, to review now, the fact that intensive and dedicated research to this disease has given us some important clues about the cause of the condition. One thing I haven't emphasized so far is that we know the immune system is involved in this disease. I guess the term autoimmune might imply that. But it's not been known for sure which of the many types of immune cells are involved or causative for the tissue damage that can occur. Beyond the cells, we've also been very interested in knowing what are the individual molecules, the proteins, that these cells might make or use to cause tissue damage. And a third major question arises from a fact that I mentioned earlier, that lupus is a heritable condition. So what are the genetic factors, and more importantly, how do they work to cause increased risk of lupus? The University of Minnesota has had a long-standing program dedicated to researching all three of these questions, starting about 15 years ago with the program headed by Dr. Timothy Behrens. Dr. Behrens worked at the University of Minnesota for over 10 years and departed in 2006. But during the time he was here, his group made some seminal observations about the molecules and the genetic factors that might increase lupus risk. Next slide. One of the principal molecules or proteins to be associated with lupus over the past decade is a group of molecules we call type 1 interferons. And we abbreviate interferons with the three-letter acronym IFN, as you see on this slide here. Interferons are cytokines. These are molecules or proteins which communicate between different cell types in the body. So they're produced by one cell type, and they are sensed, or they talk to a different cell type, or sometimes the same cell that made them. But the idea is that they are communication proteins. And the result of their communicating between cells is that the body is better able to combat the phenomenon of viral infection. I'm sorry. They have been known, they're not new. They've been known since the dawn of molecular research. About in 1957, the first report of these molecules was made in the literature. During our talk, you'll hear me say at times words like interferon alpha and interferon beta. These are just two of the members of this large family of proteins that comprise several dozen known molecules, but all have the same biologic effect as far as we can tell, in that they interact with a receptor on cells and drive that antiviral host defense program in the body. So there's a little cartoon here over on the other side, the right-hand side of the slide here, which depicts the infection-sensing cell. This would be a cell in the lung or a tissue that meets up with a virus, and upon seeing a virus, produces type 1 interferon. The type 1 interferons then interact or signal to additional cells in the body, which help the body to produce sorry, do I have control of the mouse?
Yes,
Sure.
I can take it back.
Okay, the mouse is gradually coming under my control here. I'll let the mouse go.
Okay.
So, next slide. This just gives you an idea, a little bit of the fact that we've been interested in the possibility that type 1 interferon might intersect with lupus pathogenesis, or the cause of lupus, for many years. In fact, it was in 1979 that elevated levels of interferon were measured in lupus blood, in some patients' blood. About a decade after that, investigators in Sweden documented the first evidence in 1990 that patients who were treated for other diseases, such as viral infection or multiple sclerosis, with interferon alpha, rarely could develop conditions that looked like lupus, in that those conditions had autoantibodies and other evidence of autoimmunity. So there was a hint that interferon might play a causative role in lupus as early as 1990. About a decade after that, there was additional discovery that the immune system, in the form of antibodies, could induce type 1 interferon by lupus or even healthy cells. And so the idea that interferon could be connected to the autoimmune response causally was established about that time. Exactly 10 years ago now, the first reports came out of association between lupus and expression in the blood of sometimes hundreds of genes that are controlled by type 1 interferon. This was a very important discovery in lupus history, or research history, and the first name on the slide you see there, Behrens. Tim Behrens was an investigator at the University of Minnesota at the time that he published this report, along with others at institutions across the country. One of the trainees and proteges of Dr. Behrens, a researcher by the name of Dr. Emily Gillespie, who remains at the University of Minnesota in our rheumatology division, later linked not only genes associated with type 1 interferon, but also proteins called chemokines in the blood that are driven by type 1 interferon with lupus. And that the papers describing the chemokine signature appeared in 2006. Next slide. So what is this interferon signature? As I alluded to, there are sometimes hundreds of genes that can be detected after a cell has been stimulated with type 1 interferon. And a collection, a subset of these genes are characteristically associated with lupus and are found to be increased in lupus patient blood cells. So this subset represents the interferon signature. And the signature was defined and codified, as I said, in 2003 by UMN researchers. Initial discoveries suggested that this interferon gene signature might correlate with disease activity or even specific manifestations. However, ongoing research has found that this is a complex relationship, that the interferon gene signature is in fact quite persistent through time, whether the lupus patient is in remission or is flaring badly at any given time. The type 1 interferon signature seems to remain stable. Although there is some evidence that the chemokine signature could correlate well with disease activity. The importance of this observation is that perhaps measurement of one or more chemokines in the blood of lupus patients might help doctors or providers to predict or catch early an oncoming lupus flare and adjust therapy early in the course of a flare. The idea would be, of course, to try to nip such a flare in the bud. Next slide. Here's just an example of some of the complex data sets that allow researchers to talk about the interferon signature in lupus. This display is called a heat map, and you can see across the top there a lot of vertical lines, each leading to a column, and the column contains a number of colored squares. The three major colors you might see are blue, yellow, and black. I hope the colors are the same on your screen in these columns. So each column has many little boxes, and the boxes are arranged into rows. And each row represents a single gene. So this is an experiment where blood of lupus patients or healthy controls, you can see there are 81 patients represented on this heat map and 41 non-patients or controls, have been compared for their blood gene expression profile. Yellow represents genes that are up, blue represents genes that are down, and black represents genes that are unchanged from controls. And so what you see is down near the bottom of this heat map a big blob of yellow affecting about 2/3 of the lupus patients. You can see at the top that the lupus patients are indicated by the black columns and the controls are indicated by the gray columns. So almost all of the yellow splotch down near the bottom there is associated with the lupus patients, and none of that yellow splotch is associated with the controls. All of those genes represent such genes that are known to be induced or triggered by the action of type 1 interferon in blood cells. And so this was some of the direct evidence or raw data that convinced us that interferon-driven genes are abundant in the blood of lupus patients, and it raised the hypothesis that type 1 interferon might be a major driver of disease activity in lupus. Now that hypothesis has been addressed intensively over the past decade in both animal models as well as human studies. A number of the animal models of lupus that we have available do support the idea that the type 1 interferons are important for driving, for example, the kidney disease, the joint and skin disease that is observed in lupus patients. There are examples of animal studies that counteract the hypothesis as well. So there's some, I would say the preponderance of the animal data suggests that type 1 interferon is a promoting factor in lupus in animals per se. Next slide. Here's a model that's prepared by my colleague, Dr. Emily Bachelor, formerly a student with Dr. Timothy Behrens and now a faculty member here at the U of M, in which Dr. Bachelor puts out a model for how type 1 interferon might be driving this process. Is it possible for me to try to get the pointer back again? All right. Yes, I'm moving it now. Okay. So if you look near the top of the slide, we see the inciting event is imagined to be an infection, likely a viral or a bacterial infection. And as we mentioned earlier, there are cells in the body whose purpose it is to sense the presence of infection. In this case, a group of cells called plasmacytoid dendritic cells are featured. These plasmacytoid dendritic cells are factories of type 1 interferon. They have about 1,000 times the capacity of other cells in the body to produce one of the interferons, interferon alpha, in response to infection. Sorry. I don't see the pointer anymore. I'm not sure what you all are seeing. And so type 1 interferon is thought to be produced by plasmacytoid dendritic cells and then act on other cells in the blood called monocytes. These monocytes mature in response to interferon and produce molecules that drive inflammation. And the names of these proteins are not particularly important. It's just important to know that inflammation, which is a process in the body that can result in tissue damage, is promoted by these proteins. So they are downstream of the type 1 interferons in this model. Not only, I'm sorry, did I do that?
I'm not certain here.
So in addition to the inflammatory proteins, the type 1 interferons can also cause activated cells to engage lymphocytes. Lymphocytes are important because they are capable of amplifying and augmenting inflammatory responses, which ultimately reduce or cause tissue damage. Two of the main lymphocyte subsets are depicted here, including T cells and B cells. And they are depicted interacting directly with that activated dendritic cell because the T cells and the B cells work together in a very well described fashion to produce autoantibodies. The autoantibodies in turn float in the blood, and they can lodge in places like the joints and the kidneys and induce further inflammation, which results sometimes in permanent tissue damage. These antibodies can also form a positive feedback loop. What that means is the antibodies can, just like the infections, activate the plasmacytoid dendritic cells to produce more type 1 interferon. And so the cycle goes. And this is thought to be how infections might actually induce worsening or flares of lupus, because lupus may be established already, but a new infection brings in more type 1 interferon and drives the cycle around such that disease activity is worse in the course of the infection. Okay, next slide. Maybe since are there any questions that are coming up, Cheryl? Or
Not quite yet. I'm asking the audience to do that. As soon as that happens, I will definitely kick in. So Dr. Peterson is asking for questions, please.
Okay. While waiting, I'll go into a brief discussion of this idea, this hypothesis that type 1 interferon is causative in lupus. And I can tell you that there's a very strong interest in this hypothesis, such that several major pharmaceutical companies have produced specific antibodies which block the effect of type 1 interferon. And these drugs have been and remain in testing for the last couple of years. In fact, there were some publications in our rheumatology literature just in 2012, which reported on the early safety-defining features of these anti-interferon drugs. And the good news is that these drugs indeed are very well tolerated. Furthermore, the drugs are thought to suppress the blood interferon signature. In other words, if you look in the blood of lupus patients treated with those drugs, their blood interferon signature goes away. So it seems like the drugs are working to antagonize the interferon signature. Now, we don't have any data on the question that's of great interest to patients and providers everywhere, that is the effectiveness of the drugs in managing lupus. And those findings are expected to be reported by the companies later this year. But the preliminary data suggests that a subset of lupus patients can respond to anti-interferon agents. Not everyone, and it's not quite clear why some patients respond and others don't. But there is an apparent subset that can be defined by cytokine levels and antibodies in the blood, which are likely to benefit from this managed treatment of anti-interferon drugs. What that means is not everybody's benefiting, and there's likely to be, even when the dust has settled and we know that some people benefit, it's not going to be a panacea, anti-interferon. There will still be a need for additional therapeutic approaches. So let's go on to the next slide.
Dr. Peterson, I do have a question for you. The question is, how close are those anti-type drugs available to the public? So how soon do you think they will be available?
They are not close to FDA approval. So even though the safety testing has been done, my impression is that there will need to be at least a year of additional study by the pharmaceutical companies in large trials before the FDA will look at the data to determine whether there's enough of a benefit to approve them. So in my view, we are looking at least a year before the FDA rules on anti-interferon in lupus.
Awesome. I have one more question for you. Please discuss the current clinical trials with IFN, and are human beings being recruited? Where can we read more about this research?
Yeah. I don't have the websites or the direct contact information right at hand, I'm sorry. But there are the companies that are making the anti-interferon drugs and testing them are a company called Genome in San Francisco and another company called MedImmune, which I believe is based in Massachusetts. And I think if you were to go to these company websites and type in type 1 interferon or anti-interferon, you'd be able to gather information about the trials. I'm sorry, I don't know where these trials are recruiting patients or if they are still recruiting patients at the moment. As far as I know, both agents from each of these two companies are in independent trials, however. All right.
Oops, I'm sorry. Okay.
So, what can we do for those patients who unfortunately may not respond to anti-interferon therapy? Well, I think we need a new pathogenetic model for how type 1 interferon works in some lupus patients for those people, because it's clear that these anti-interferon agents are actually blocking type 1 interferon, yet if the patients aren't getting better, it means then that the drug is effective to do what it's supposed to, but it's not helping the patient. We need a new model for moving forward. And to answer this, so to look further, what we've done is gone back to the results of very powerful genetic studies to get some clues as to how to help these anti-interferon non-responders. Next slide. So we've been interested recently in this last question. How do the genetic factors control the cells and molecules that drive lupus? Next slide. Just to remind you, genes do matter in lupus. If you are a person who is a first-degree relative, a sibling or parent or child of a lupus patient, you bear about a 3 to 5-fold increased risk of developing the disease yourself at some point. if you are the identical twin of a patient with lupus, you have about a 20 to 30-fold increased risk of developing disease. So these observations strongly suggest that lupus is in part a heritable condition. Next slide. Now, in the past decade, there have been, I think, seminal discoveries concerning individual genes amongst the tens of thousands of genes that we have in our bodies, individual genes that predispose people to lupus. And this research has been made possible because of technological advances in the ability to sequence large tracts of human DNA rapidly and accurately. It's worth knowing that each of us has enough DNA in every one of our cells that if you lined up the DNA and strung it out, it would go from here to the moon. And so there's a lot of it. 3 billion pieces of information in each of our cells' DNA. You might think about this collection of DNA in each of our cells and bodies as a map to the genes, because the genes are located in this collection of DNA. And the genetic map, as everyone knows, varies slightly between people in hundreds of thousands of locations. And these sites of variation can be detected by sequencing, determining the primary sequence of the DNA, and they're called single nucleotide polymorphisms, or SNPs for short. In my mind, I view these SNPs as signposts or location markers that we can use to search for the location of genes that influence the course of lupus. And you can see depicted on the right hand of this slide a single base pair in that beautiful double helix, which represents DNA in our bodies, where in a healthy person there's one base pair in the DNA that in a lupus patient is changed. And so it's a polymorphism or a SNP. Next slide. I just want to emphasize that our knowledge of the human genome has advanced exponentially in the past decade. In 2002, if you were to think about the map of the human genome and analogize to what was known about, say, the North American continent, you'd think about a map that is depicted on the left side here. It was a very broad and crude knowledge of the human genes. And we mainly didn't know how genetic maps differed between individuals. So you might have said that in 2002 we knew the location of features like rivers and mountains, but not of individual towns, roads, or state boundaries. So the next slide. Since then, however, again, technological advances have allowed us to specifically identify over 400,000 places in the genome, individual signposts that vary from person to person. And using powerful computers, we're able to compare those signposts in the DNA of thousands of lupus patients and thousands of control subjects and compare them. We can generate a signpost map, as it were, of the whole genome for every person based on these SNP locations. And a typical research technique is that researchers then compare the maps of individual patients and individual controls and come up with likelihoods or statistical tests that tell us where it's likely a particular or a hot spot, meaning a place where there are many signposts associated with lupus patients but not controlled, not healthy subjects. I hope this makes some sense.
I do have a question for you, Doctor.
Yes.
The question is, with so many, as you call them, signposts, is that why diagnosis of lupus is so difficult?
No, I don't think so. Although I think that as I'm going to mention in a minute, this whole genome association research has resulted in the definitive identification of over 40 different genes that can predispose people to lupus. What that means to me is that there are probably many different ways that a person can come to a lupus diagnosis. And so now I'm coming around as I'm talking to agreeing with the questioner that yes, probably the results of these studies suggest that many different genes are involved in causing lupus. There are many different pathways resulting from those genes being activated. And so it makes it hard for practitioners to get a handle on all of those genes and lump them all together in a single diagnosis of lupus. Next slide.
I do have one more question, if that's okay. With
Yeah.
genome mapping that was done 10 years ago, is the secondary analysis of these SNPs, or I'm sorry, is the secondary analysis of this SNPs, and what is the likelihood of discovering causes and cure for more rapidly by focusing on these signposts?
Yeah, well that's what I'm excited about, is the next step. So as I said, these genes, in October 2012, there was a report in the literature of 47 definitively described risk alleles or risk genes for lupus. Of those, complete understanding of how the genes work exists for 0. So we have some clues about the function of some of those genes in lupus, but the next great leap forward will come from detailed molecular analysis of each of these genes to try to understand how they can be linked to the autoimmunity that characterizes lupus. And when the secondary analysis question, additional genome-wide association studies with even more thousands of patients and more thousands of controls are being done. But it's my impression that the geneticists are now using even more advanced sequencing technologies to rapidly sequence entire genomes of individuals within 48 hours and use those maps to find what are known as rare variants. This technique can be applied usefully to families in which lupus is known to run with great frequency. And so comparing the genome of a parent with lupus with the child of that parent who has lupus also, and finding which of the genes are shared gives more clues to what might be happening in lupus. And so the next, I think, wave will come from identifying rare variants in genes that don't show up on these big whole genome association study radar screens. Next slide. This is, again, just a visual depiction of how I imagine that these hotspots might be appearing in the more detailed map of the human genome that we now have available, which wasn't available in 2002. I want to spend a little time now talking about one of the first fruits of these genetic studies. This is a gene called PTPN22, a known lupus risk gene. PTPN22 is a gene that encodes a protein found in T lymphocytes as well as dendritic cells, other white blood cells in the body. And it's fascinating to me that a single amino acid chain, there are 800 amino acids in this PTPN22 protein, and one single change, 1 out of 802, is enough to cause the increased risk of lupus and other autoimmune disease. So carrying this single amino acid change is a very powerful risk factor for lupus. It's also interesting to note that Minnesota is enriched, is a great place to be if you are interested in studying how PTPN22 contributes to lupus, because it's a gene that's found in people of Northern European and Central European extraction. And in contrast, if I had a laboratory in Uganda or in Hong Kong, it wouldn't be a very good place to study PTPN22 because African Americans, Africans, and Asians have a very low incidence of this gene. Okay, next slide. Just a brief overview now. How do we go about studying the question of how PTPN22 might increase lupus risk? And here I'd like to emphasize that we've used parallel corroborative approaches using mouse models of disease as well as direct studies in human cells. And I'd like to spend a little time just talking about the mouse models first. So using technology that allows us to change the chromosomes of mice, we've been able to generate animals that either lack the PTPN22 gene entirely, or animals that are programmed to express the human form of PTPN22 that's associated with lupus. I hope that's clear. So it's a mouse that is furry and runs around the cage in every other way next to its mates, but a mouse that carries a human form of a single human gene, and that gene is the PTPN22 gene. How does this help us? Well, it helps us because it allows us to study animals that are essentially identical twins, but hundreds of them. And it's very hard, of course, in humans because we are very genetically heterogeneous, very distinct. It's hard sometimes to parse out the effects of a single gene in a very complex genetic environment like humans. So the uniform genetic environment of animals is a great benefit of using them for these studies. And so we've used a mouse in which PTPN22 has been knocked out, is the jargon we use. The gene has been deleted from the mouse. Every other gene in the mouse is normal, and you can't tell looking at a PTPN22 knockout mouse sitting next to its normal litter mate in the cage which one lacks PTPN22. One has to do sophisticated analysis of the DNA from the knockout mouse to show that the gene is gone. We have tested these mice for various forms of lupus inflammation, and we have found, interestingly, that PTPN22 is a major factor in controlling the severity of experimental inflammation. That experimental inflammation is dependent upon, of all things, type 1 interferon. A second initiative or parallel approach to studying PTPN22 in lupus has come from our work with human cells. We have asked the question, does PTPN22 affect the behavior of white blood cells when they're responding to infection stimuli? Just as I showed you in the model by Dr. Gillespie earlier in the presentation, we can take blood cells from people and give them substances that mimic viral infection and then monitor their activation, their behavior in terms of the genes that and proteins that they produce. And so what we've done to date is identify people, in this case healthy people, who carry the lupus-associated PTPN22, or people who don't carry the disease-associated PTPN22. We bring them both into the lab, draw their blood on the same day, stimulate their blood cells with the infection mimics, and monitor the immune responses. And what we found is, in some, that the carriers of the lupus-associated PTPN22 form have a very different response to infection mimic stimulus. And that different response reflects type 1 interferon. Now, the bottom line is PTPN22 risk carriers have less type 1 interferon than non-carriers. I'll repeat. The white blood cells of PTPN22 risk gene carriers have less type 1 interferon than those who are non-carriers. Next slide. This slide, again, just summarizes what I've been telling you, that when we look in the mice, that lack PTPN22 on the left, we find that those animals cannot suppress a form of inflammatory arthritis that mimics a kind of inflammation that we see in people with lupus. And when we look at the human cells, we find that those cells are weak responders to infection stimuli in terms of type 1 interferon production. So we conclude from these studies that PTPN22 influences inflammation and type 1 interferon responses to infection. Next slide. And so here's a model that I've created that just summarizes pictorially what our research has shown about PTPN22 so far. And by the way, I guess I've been showing you a protein on the slides now called SLE lip, and that refers to PTPN22 by another name. Lip is the abbreviation for the protein form of PTPN22 gene. So the lupus-associated PTPN22, or SLE lip, we find is associated with reduced production of type 1 interferon. You can see this schematically on the left side of the slide where normal lip is found in a cell. A microbe such as a virus or a bacterium comes along and engages a receptor on that cell. And normal lip is involved with promoting the production of type 1 interferon on that cell. Okay, you see all the teardrops that are labeled IFN in the bottom. That's all the gobs of interferon produced in a cell bearing normal lip. On the right-hand side of the slide, we'll contrast now. The microbe comes along, hits the receptor, drives a signaling pathway to produce type 1 interferon. But because the SLE lip is present in that cell, there is less type 1 interferon coming out of the pipeline at the end. And so the signaling pathway is not promoted as efficiently when a person carries this SLE lip. And the end result is reduced type 1 interferon. This is a critical point. This is our main observation about this gene. Next slide. So another way of pictorially presenting this is that normally these infection-sensing cells in our body, which we call myeloid cells, produce type 1 interferon after they encounter a virus or a microbe, shown on the left. And when they produce the type 1 interferon has several major effects on the body. The first effect would be what I've described already, that interferon is one of the fundamental building blocks of our ability to fight viral infection. It's a phenomenon we call host defense. A second major function of type 1 interferon, though, that's increasingly recognized is the ability of type 1 interferon to suppress or antagonize inflammation. And so in a normal myeloid cell carrying the non-lupus associated lip, when it's stimulated by infection, it produces interferon, and that lots of interferon may actually help to prevent inflammatory tissue damage. That's depicted on the lower right-hand side of the slide. And so PTPN22 is acting in this signaling pathway within a myeloid cell, but its effects downstream are very widespread in terms of antiviral host defense as well as a blockade of inflammation in the tissues. Next slide.
Dr. Peterson, we have a poll to launch, so I'm going to launch that right now.
Okay.
And there it is.
Yes, so this is just to make sure we're all on the same page about what I'm thinking about type 1 interferons.
And we'll give it a few more seconds.
All right, is the poll complete?
We have, I think, about 2 seconds, and then I will close it, and I'll give you the results. All right. So the consensus has been all of the above.
Were there any that liked any of the individual responses more than the others?
20%. The other, it's actually 20% went for they are important for fighting viral infections.
Okay.
Otherwise, 80% chose all of the above.
Well, I applaud those who pick out the fighting viral infections as the best described and well-established function of type 1 interferons, but the answer actually is all of the above. Okay, so next slide. This slide, again, I think is now depicting what might be occurring in the situation where the myeloid cells in a person are not carrying the normal lip, but instead are carrying the lupus-associated lip. And you can see the little X's at all the various arrows connecting production of type 1 interferon to host defense and inflammation. And in general, the end result of carriage of this variant lupus gene might be uncontrolled inflammation that could enhance the risk of tissue damage in lupus. Next slide. So what this has done is set up a kind of paradox. As I said earlier, most lupus patients have evidence of too much type 1 interferon in their blood. But our results with PTPN22 suggest that the carriage of the lupus-associated PTPN22 protein results in too little type 1 interferon production by the blood cells. How could this be? Next slide. Well, our current model is, and this is a model that's in dynamic, it's not complete, but it could be that the cellular source of interferon could explain how some lupus patients have disease because of overproduction and others have disease because of underproduction of interferons. In the case of body-produced type 1 interferon, too much of the type 1 interferon made in these tissues, the kidneys, the lungs, the heart, the joints, could cause autoimmune damage to joints and skin. In the case of blood cell-produced type 1 interferon, and this is the type that I would imagine is happening with PTPN22, too little type 1 interferon could result in worsened or increased susceptibility to infections and therefore more inflammation-associated tissue damage when the body tries to respond to these infections with inflammation. I will not go into any more detail about the model right now because I want to take a few minutes at the end here to describe a study that will help us understand this better. So just to show you, again, the timeline, but now the study. And this is a study for which recruitment is now ongoing. The major question or goal of the study is to ask, how does a lupus-associated form of PTPN22 affect the immune cell ability to produce type 1 interferon? What are we looking for? We are seeking lupus patients. They can be ages 18 to 80. They should be female. And they should be Caucasian. As I said, the lupus, this is not because of racism. This is because the lupus gene variant is found in only a very small fraction of Asian or African American people who incidentally get lupus, of course. But we think that the mechanism of lupus may be very different in these patients because of this PTPN22 study. We need to have evidence that the lupus diagnosis has been established by a rheumatologist. And anyone who'd like to participate in the study should be willing to donate a tablespoon of blood and to be contacted later, once we have used that blood to establish the genotype of the person, to consider donating an additional 4 tablespoons of blood so after the genotype is confirmed. Next slide. So again, we're looking for persons with Northern or Central European descendants or ancestors who have lupus. And I put the slides of the Scandinavian countries on the top there because the incidence of this lupus-associated gene is the very highest in the world in those countries of Finland, Norway, Sweden, interestingly. Next slide. So we're coming down to the last few minutes here, but I wanted to just briefly review what we've talked about. the disease lupus remains incurable, but I'm excited that both our understanding of the disease cause and new treatment options are improving and on the road. The studies of lupus genes like PTPN22 have pointed to important new research questions and directions. And this molecule, type 1 interferon, is definitely wrapped up with the cause of lupus. Whether too much or too little might predispose to lupus is still an open question, but interferon is an important driver. And we've got a new study to plumb this question further. This will use, the idea of the study is to test the interferon-producing capacity of white blood cells in lupus patients themselves. Previously, we've studied only healthy humans who carry the lupus gene. Now we'd like to look in lupus patients themselves. Next slide. A grateful shout out to the Lupus Foundation of Minnesota, not only from me, but from other researchers and staff at the University of Minnesota. Pictured here at the top is the division director of the Division of Rheumatic and Autoimmune Diseases, Dr. Daniel Miller, and my colleague, Dr. Emily Gillespie, or Bachelor. Her married name is Gillespie, but her publications are sometimes under Bachelor. And she's one of the people who set us on this road to studying type 1 interferon in the first place. So I think that's the last slide I had. Oh, just to tell you a quote that I like from Dr. Osler, who is a very famous internist in the early part of the 20th century. Why we get up and do research in rheumatic disease? Well, we want to wrest from nature the secrets that have perplexed philosophers in all ages, to track to their sources the causes of disease. These are our ambitions. So that's why we get up in the morning. All right, and we're not content with the status quo. Are there other questions I can answer now?
I do have a question for you. Is there a deadline to enroll in your study?
We haven't set a formal deadline. The enrollment is already in motion. We are aiming to enroll approximately 150 subjects with lupus. Currently, we have enrolled 37 subjects after approximately 6 weeks of beginning to enroll. So I anticipate that our enrollment will go on for at least another 4 or 5 months. But I would say if you are interested and want more information, I'm happy to provide it quickly right away so we can get you into phase 1 of the study.
And you'll notice on the slide, there's the contact information for Valerie Furman. She is the study coordinator. Her preferred method, if you are interested in joining the study, or if you are a provider and want to share with your patients in case they may want to also become a part of the study. She does prefer email, but otherwise her telephone number is there, and she asked us to take both of those contact methods down. Okay. I think we are right at the end, Dr. Peterson. So on behalf of the Lupus Foundation of Minnesota, I want to thank you for taking the time from your busy schedule to provide us with today's presentation. So thank you so much, Dr. Peterson.
It's been my pleasure. And again, I'm willing to be contacted by any viewers who had any unanswered questions or had further interest in the topic.
Absolutely. I also want to thank everyone for joining us today. We hope that you really enjoyed today's webinar. I do apologize for some of those technical glitches that we talked about earlier. You will receive a survey via your email, so we ask that you complete it as we use this feedback to improve programming as well as offer new programming. In case you missed the information during the presentation and you are interested in becoming a part of this study, we would like you to share that information with your patients. That information will also be contained in the survey, so you can jot it down quickly. And if you need a certificate of attendance, please feel free to email me at the address on the screen, and I will get that out to you before week's end. So again, thank you everyone for joining us, and have a great afternoon.